Hey everyone, and welcome to part 2 of our week 2 presentation covering the urinary system. So we've already gone into some of the anatomy of the kidneys, and now I want to talk about the functional unit of the kidneys in greater detail. And by functional unit, I mean the part of the kidneys that's actually doing the filtration of blood. So the functional unit of the kidneys is the nephron, and this is what I've drawn here as one example of one nephron. Again, remember, each kidney is going to contain between 250,000 to a million nephrons. So there are many of these nephrons in the kidneys. And what you should notice, you may recognize this structure that I've drawn here at the start of the nephron. So these tiny arteries that are supplying the blood to be filtered are going to pass blood into the glomerulus. This is the renal corpuscle, which contains the glomerulus, which are those capillaries inside of the Bowman's capsule. So Bowman's capsule surrounds the glomerulus. And this is where ultrafiltration of blood takes place. And ultrafiltration just means to filter out things that are pretty small. So blood that's filtered, or the filtrate that's filtered out of blood, is going to enter the nephron. And the nephron has three main regions. So here, this first region of the nephron that the filtrate is going to pass through is the proximal convoluted tubule. So this whole nephron is just a series of tubes or tubules, and so we describe these tubules as being either proximal or close to the glomerulus. There's also a distal convoluted tubule, and that term convoluted just means kind of twisty. So the convoluted tubules kind of twist around. So there's a proximal one that's close to the glomerulus, a distal convoluted tubule that's further away from the glomerulus. And then you have the loop of Henle. So one important thing to note about the nephron is that it's going to cross the boundary between the renal medulla. I've actually got these mixed up. Sorry about that. So mix those up. This is the cortex. This is the medulla. So when we looked at those renal pyramids before, those little triangles, those basically form the boundary between the two prime regions of the kidney, which is the outer cortex and the inner medulla. So the nephron and the glomerulus are going to be primarily located out or completely located out in the cortex. And the proximal and distal tubules are going to be located in that cortex as well. The loop of Henle, which consists of both the descending limb, so there are two limbs, a descending limb that descends down into the medulla, and an ascending limb that ascends or climbs upward back toward the cortex. So the loop of Henle is going to dip down into the medulla. And remember, the medulla is going to be a very salty environment. So there's lots of salt out in this region. And that salt is going to draw water out of those tubules by osmosis. So that's going to be one of the main ways that fluids are able to return back to the circulation, is osmosis that occurs in the medulla, in this loop of Henle. So the nephron that I've drawn here, I've also given you some arrows that represent the flow of material. So the first flow of material that we have, or passage of material, is filtration in the glomerulus, this blue arrow. So filtrate is removed from the blood That's going to contain things like water, of course, nutrients, your glucose, amino acids, electrolytes, waste. And that filtrate is going to pass through the tubules. And as it passes through the nephron, there's going to be movement of the substances. And substances in the filtrate can be reabsorbed. So these green arrows that I've drawn along the path of the filtrate are going to be places in the nephron where substances can be brought out of the filtrate. So whatever was removed from the blood, that includes some things that we actually want to return to the blood. And so those substances, like your glucose and amino acids, that are really useful to have, are going to be reabsorbed instead of passing through the whole tubule and out the collecting duct. So the final part of the nephron is this collecting duct. And that's where anything that hasn't been reabsorbed is then going to be collected and passed on toward those calyces and eventually toward the ureters as urine. So the collecting duct just collects all of the substances that we want to remove from the body. So as well as things being reabsorbed that are useful, there's also a lot of water that's reabsorbed. Things can be secreted. They can go the other direction. So reabsorption is material that's passing from the filtrate back out to the blood. And how this is possible, one structure that I haven't drawn here are the peritubular capillaries that I mentioned previously. So here's the afferent arteriole that feeds blood into the glomerulus. Here's the efferent arteriole that carries the remaining blood away from the glomerulus. And that's going to then transfer into a network of peritubular capillaries that are just going to surround. They're going to make basically a web around the nephron. So all around the nephron are these peritubular capillaries that are going to contain blood. And so that blood can reabsorb things that are useful, like those nutrients. Certain electrolytes can be reabsorbed. Water can be reabsorbed and put back into the blood. And so in that case, reabsorption is occurring, materials moving from the nephron back to those capillaries that contain blood. So that's how material gets back into the blood. Secretion is just the opposite of that. Those peritubular capillaries still might have some waste that might not have been filtered out at the glomerulus. And secretion is going to be when material moves from those peritubular capillaries into the nephron. So things that are secreted from the blood are things that we want to get rid of, that we want to put into the nephron that's going to make it so that those substances make it out into the urine and are removed from the body. So these three forces are going to control and be able to fine-tune exactly what the chemical composition of your blood is. And that's very important for maintaining homeostasis of all sorts of different biological variables, from blood pressure to electrolyte balance to pH, those things that we mentioned yesterday that the kidney is really important in controlling. So what you may also notice about this nephron that I've drawn is that there's some places where the nephron appears skinnier than others. And the reason for that discrepancy, the difference in the size of different parts of the nephron, is because the cells of the walls of these tubules are different in different places of the nephron. So a couple of different types of cells that you may remember, and these are epithelial cells, these are both going to be simple layers of cells, so single layers of cells. So in the places where the tubules are thick, those are going to be simple cuboidal cells. Simple because they're a single layer, cuboidal because they've got kind of that square shape. Whereas in the thin regions of the nephron, you're going to have, again, a single layer of cells, but they're going to be those flat, thin, stretched out cells, your simple squamous epithelium. And along with there being differences in the shapes of cells, so you have cuboidal that makes these thick portions, squamous that makes these thin. Those cuboidal cells can also have microvilli. So what you will hopefully remember about microvilli is that they are extensions of the cell just designed to increase the surface area of those cells. So cells that have microvilli have more plasma membrane that's exposed to the substances that are adjacent to it. So why this is useful, particularly in places where there's lots of reabsorption occurring, is the more surface area you have, the more capacity you have to absorb things or to reabsorb things. So in this proximal convoluted tubule, there's lots of reabsorption going on. So let's think about what's making it out into this proximal tubule. Well, when you first filter the blood through ultrafiltration in the glomerulus, you're going to have lots of nutrients that we want to reabsorb as quickly as possible. We don't want our nutrients, in a healthy individual, you're not going to have things like glucose and amino acids that are present in high amounts in your urine. And that's because your body can use those. There's not a good reason to remove those from the body. Now, someone who's diabetic who can't control their blood sugar is going to lose a lot of sugar in their urine. But that's an unhealthy individual. So in healthy people, you are reabsorbing most of your nutrients immediately in the nephron. And so the way that this is possible, structure relating to function, is that these cuboidal cells in the proximal convoluted tubule have a dense layer of microvilli. They have lots and lots of microvilli. And when you have lots of microvilli, anatomists call that a brush border. It kind of looks like a hairbrush, I guess, with the microvilli being analogous to the bristles on a brush. So it looks kind of bristly. But those bristles are just going to allow these cells to do much more absorption than they would if they had fewer microvilli or no microvilli. Now, as you move on to other regions, you see there's still reabsorption going on where you have these simple squamous cells, but it's different type of reabsorption. So in the loop of Henle, the substance that's going to be reabsorbed the most is water. And again, that relates to what exactly the nature of the medulla is. It's salty. It promotes water reabsorption. So water is getting reabsorbed here in the loop of Henle. Once you move on to the thick ascending loop, or thick ascending limb of the loop of Henle, you get a transition back into simple cuboidal epithelia. But these epithelia here, in the thick ascending limb, contain no microvilli. They do contain sodium pumps, so you're actually going to be doing some reabsorbing here, but not because there's microvilli that are particularly good at absorbing things. It's because there are actually pumps on the membranes, and we'll get more into that in the future. But largely, this thick ascending limb is impermeable. It's impermeable to water. There's not a lot of water reabsorption going on in the ascending limb. But again, the simple cuboidal epithelia in the ascending limb contains no microvilli. So there's much less reabsorption going on here than there is here in the proximal. And then once you get back into the distal convoluted tubule, there actually is still some reabsorption going on, but the simple cuboidal epithelia here in the distal tubule are going to have fewer microvilli than in the proximal convoluted tubule. And this relates to, you could probably imagine, go ahead and imagine that, or you might guess that the distal tubule is doing less reabsorption than the proximal tubule, and you'd be correct. The distal tubule and the collecting duct are both going to be places where fine-tuning occurs. So the amount of reabsorption and secretion that's going on in these two places is actually going to be dictated by the levels of certain hormones or chemical messages are going to adjust, fine-tune how much reabsorption is going on in the distal tubule and the collecting duct. So here in the proximal tubule, your nutrients and things are being reabsorbed the same all the time, no matter what the circumstances are. You're usually always going to be reabsorbing nutrients and things like that. But in the distal tubule and the collecting duct, again, they're under the influence of hormones. So certain hormones have to be present for certain substances to be reabsorbed or not reabsorbed or secreted in the distal tubule and the collecting duct. So mainly I just want to get across with this figure the idea that the nephron and the kidney itself is designed to allow your body to specifically alter the composition of blood. So the way that the nephron can reabsorb certain substances and change the chemical composition of blood is very specific, and that's to maintain very precise range of concentration of certain substances, and also to maintain things like blood pressure and pH. So this is a really efficient tool for doing that. This is a very complex and efficient filtering system. And one thing that adds to the complexity of that a little bit is the fact that there are multiple types of nephrons. So the nephron that I've drawn here is called a cortical nephron. And it's called a cortical nephron because it's located mostly in the cortex. And the loop of Henle only dips down into the medulla very superficially. It only dips down just a little bit. So this is called a cortical nephron, and most of the nephrons in the kidney are these cortical types. The other type of nephron that you'll find has a much longer loop of Henle. So this is just my basic drawing of a long loop of Henle. But you can see that in this type of nephron, which is called a juxtamedullary nephron, juxtamedullary nephrons are going to project much deeper down into the medulla. And why they do that is because juxtamedullary nephrons are designed to maximize water reabsorption. So the fact that they exist, they dip down much further into this salty medulla, and that provides just a lot more surface area of tubule for water reabsorption to be taking place. So again, there are many more cortical neurons than there are juxtamedullary, but juxtamedullary neurons are important for allowing us to fine-tune water reabsorption. So So now that you have some information about generally what the function of the kidney is or how that's achieved using the nephron as a very specific mechanism for filtration, I want to talk about the filtration membrane for a minute. What is actually the membrane in the glomerulus that allows certain substances to pass into the nephron but keep others into the blood? So if you take a look at the glomerulus, which again is a set of capillaries inside the renal corpuscle. So the renal corpuscle again is this glomerulus, these capillaries that's fed by the afferent arteriole, and also the capsule, Bowman's capsule that surrounds the glomerulus. And that's going to be where the filtrate moves into the nephron. So if you take a closer look at these glomerular capillaries, you'll see what should look like a familiar blood vessel. So what I've drawn here is just a blood vessel in red, and those little red dots that I've drawn are fenestrations. So these capillaries are fenestrated. That's one type of capillary that we discussed before when we were talking about the circulatory system. So there are tiny holes in these capillaries that allow some material to pass through. But those holes are going to be too big to keep certain things in the blood that we still want there. So another mechanism, an addition to this filtration membrane, is going to be from what are called podocytes. So podocytes are going to be cells that wrap themselves around the glomerular capillaries. So the term podocyte actually means a cell with a foot, basically. Pod means foot. And they have these little feet-like projections called pedicels. And those little projections are going to kind of wrap around the glomerular capillaries, and they're going to make little filtration slits. So those are going to be even smaller, those slits are, than the fenestrations of this capillary. And that's going to allow much finer filtration. So the smaller the holes that you're pushing filtrate through, the more things are going to be retained in the blood. And there are certain things that we definitely want to keep in the blood. We don't want to release that into the nephron. And the things that we keep in the blood in these glomerular capillaries, things that won't pass through that filtration membrane, are going to be your white blood cells and red blood cells. Of course, those are very big. Also your platelets, and some proteins are also going to be kept in the blood, are not going to be able to fit through this filtration membrane. So albumin, what you'll probably remember from our discussion about capillaries previously, is that albumin is kind of a big protein and there's lots of it in the blood. And this albumin, it's necessary to keep it in the blood because it's going to provide colloid osmotic pressure. And we'll look at why colloid osmotic pressure, or how that's going to contribute shortly. But so albumin and some other proteins are going to be kept in the blood as well. Now, things that are going to be filtered out of the blood, substances that are filtered that make it into the nephron. Of course, your water. Water molecules are very small. But you're going to also have things like waste, urea, electrolytes, amino acids, some hydrogen ions, things like that, are going to make it out into that filtrate through this filtration membrane. So this is the structure of the membrane inside the glomerulus that allows for ultrafiltration, or filtration of tiny things. So as was the case when we were talking about filtration at the level of the capillary and the tissue, filtration pressure is also important when you're talking about glomerular filtration at the glomerulus. So the net filtration pressure, which is going to determine the direction of movement of fluid either into or out of the capsule, is going to be determined by three pressures. The first pressure that's going to affect filtration is going to be the hydrostatic pressure of blood inside the glomerulus. So as blood is pushed into the glomerulus, the force that fluid puts on the walls of the glomerulus is the glomerular hydrostatic pressure. And this is, of course, related to blood being pumped by the heart. So glomerular hydrostatic pressure is going to be equal to about 50 millimeters of mercury. Now, that is a pressure that's pushing this direction, out of the glomerulus. And there are two sources of pressure that are going to oppose the glomerular hydrostatic pressure. The first of these is going to be from a concept that we've already discussed, which is the colloid osmotic pressure caused by albumin. So again, in the blood that fills up the glomerulus, there's going to be lots of albumin proteins. And these are going to tend to draw water toward them. So there's an osmotic pressure caused by albumin in the blood. And remember, albumin is too big to be filtered out into the capsule. So it's going to stay in the blood and cause water to be drawn in this direction. Now that colloid osmotic pressure is equal to about 30 millimeters of mercury. So not enough yet to really challenge this 50 millimeters of mercury. That's going to be the driving force still. The second force that's going to oppose the capillary hydrostatic pressure is going to be, or the glomerular hydrostatic pressure, I should say. The other force that opposes that is going to be the capsular hydrostatic pressure. So as fluid is forced out of the glomerulus, it's going to fill up Bowman's capsule. And once Bowman's capsule is filled with fluid that's been pushed or filtered out of the blood, that filtrate, when it fills up the capsule, it's going to cause a hydrostatic pressure in this direction that's going to oppose the movement of more fluid out into the capsule. So both the glomerular colloid osmotic pressure and the capsular hydrostatic pressure are going to oppose the glomerular hydrostatic pressure. The capsular hydrostatic pressure is going to be equal to about 10 millimeters of mercury. So if you combine these two, you'll see there are 40 millimeters of mercury of pressure that are opposing this glomerular hydrostatic pressure. And if you subtract those, you get the net filtration pressure, which is positive 10. So that means the sum of all these pressures is equal to 10 millimeters of mercury pushing fluid out of the glomerulus. So that's how net filtration pressure is calculated, and that's how we know that fluid substances should be being filtered out of the glomerulus due to those pressures. So the sum of those forces we just mentioned, leading to a positive net filtration pressure, means that glomerular filtration is going to occur. There's going to be filtration and formation of filtrate. So substances being removed from the blood, traveling into the tubules. So what that leads to is glomerular filtration, of course, and depending on the pressure, primarily the hydrostatic pressure in the glomerulus, there can be changes to the rate of glomerular filtration. So we think about the glomerular filtration rate, how much filtrate is being formed at a given time. And changes in blood pressure could cause changes in glomerular filtration rate. The higher your blood pressure, the higher that hydrostatic pressure is. And we want to maintain a normal glomerular filtration rate of about 125 milliliters per minute. So about 1/10 of a liter per minute of filtrate is being formed under normal circumstances. And there are homeostatic mechanisms in place to keep it at this constant filtration rate of about 125 mils per minute. So how do we go about affecting the glomerular filtration rate even with surges in blood pressure? Well, it mainly gets to controlling constriction and dilation of these blood vessels, the afferent and the efferent arterioles that feed blood into or take blood out of the glomerulus. So you can think of the afferent arteriole as being the faucet of this glomerulus, whereas the efferent arteriole is akin to a drain. So thinking of the glomerulus as your sink, the faucet is putting blood or fluid into the sink, and the drain is removing that fluid from the sink. So you can change how much fluid is in the glomerulus by altering either the faucet, turning it up or turning it down, or the drain, clogging it or unclogging it. And so vasoconstriction and dilation of these arterioles can achieve both of those purposes. So let's think about this. Let's say your blood pressure is high, meaning there's going to be increased hydrostatic pressure that would increase the glomerular filtration rate. So how would your body compensate to bring glomerular filtration rate back down? Well, one way that it could do that would be to turn down the faucet. And the way to do that, to slow how much liquid is getting into the glomerulus, would be to constrict or vasoconstrict this afferent arteriole. So constriction of the afferent arteriole is akin to turning down the faucet, turning down the amount of liquid that's flowing into the glomerulus. Another way to do it would be to unclog the drain or to dilate the efferent arteriole. So constriction here or dilation here would both serve the same purpose, and that would be to lower the glomerular filtration rate in the case of something like high blood pressure occurring. Now, if the blood pressure falls too low and we want to increase the glomerular filtration rate, well, of course, you would turn up the faucet. And the way to do that is to dilate the afferent arteriole, or to also you could clog the drain. Both of those, either turning up the faucet, dilation of the afferent, or clogging of the drain, vasoconstriction of the efferent arteriole. And those would both lead to a significant increase in the glomerular filtration rate in the case of something like low blood pressure. So again, dilation and constriction of the afferent and the efferent arterioles is akin to turning on or off the faucet or clogging or unclogging the drain. And in that way, we can regulate the glomerular filtration rate, keep it at about 125 mils per minute. So now I want to spend some time talking about specific mechanisms that are used to control glomerular filtration rate, again, in response to something like changing blood pressure. So there are what are called autoregulatory mechanisms for controlling glomerular filtration rate. And this is very similar to what we saw with tissue perfusion in the systemic capillaries, is that they're able to auto-regulate, meaning the glomerulus can control its own filtration rate without outside input, say from hormones or from the nervous system. So it has auto-regulatory potential, meaning it can control itself. So the first auto-regulatory mechanism is one term that you might be familiar with, is the myogenic mechanism. And what that means is it's myogenic, meaning it's going to cause a change in contractility or contraction or dilation of smooth muscle surrounding the afferent arteriole. So remember, the afferent arteriole is the faucet. Let's say we have an increase in blood pressure. You would see a corresponding increase in the glomerular filtration rate due to that increase in blood pressure. So to bring glomerular filtration rate back down, to keep it constant, the myogenic mechanism is going to detect changes in blood pressure with stretch receptors. The higher the blood pressure, the more stretched out these arterioles are going to be. So high blood pressure stretches this arteriole, and it can detect that. So what that stretch receptor is going to ultimately cause is going to be contraction turning down the faucet by contracting smooth muscle of the faucet. And what that's going to do is decrease the amount of fluid that's getting into the glomerulus. So turning off the faucet, again, is a way to lower glomerular filtration rate in the case of high blood pressure. And the myogenic mechanism can help to do that. Now, if you have a decrease in blood pressure and we need to increase glomerular filtration rate to compensate for that, the myogenic mechanism does just the opposite. So instead of turning down the faucet with a decrease in blood pressure, you're going to have to turn up the faucet. And that's when you get dilation of smooth muscle surrounding the afferent arteriole. So that is the myogenic mechanism, either contracting or dilating or relaxing smooth muscle surrounding the afferent arteriole. And that will ultimately control glomerular filtration rate. So the second autoregulatory mechanism of maintaining glomerular filtration rate is going to be through what's called the tubuloglomerular feedback system. And this is going to utilize what's called the juxtaglomerular apparatus. Now, what juxtaglomerular means is that this apparatus is next to the glomerulus. And where you find the juxtaglomerular apparatus is actually on the distal convoluted tubule. So the way I drew it previously was that you have the proximal convoluted tubule close to the glomerulus, and then far away is the distal convoluted tubule. But in real life, in actuality, and your book shows some good images showing how this occurs actually. In real life, the distal convoluted tubule loops back around and is going to pass directly by the glomerulus. So in the distal convoluted tubule, you have this juxtaglomerular apparatus that's sitting right next to the glomerulus. Now, what this allows for is a specific patch of cells in the juxtaglomerular apparatus to sense changes in glomerular filtration rate and actually have an effect on the glomerulus. So let's look at how this occurs. In the JGA, the juxtaglomerular apparatus, this specific set of cells that's going to be regulating filtration rate is called the macula densa. So these cells that I've drawn here are the macula densa. So let's think about, and this is going to, this mechanism is going to allow us to decrease glomerular filtration rate in the case of something like high blood pressure. So when you have increased blood pressure, or increased glomerular filtration rate, what that's going to do is it's going to cause an increase in the amount of fluid, the amount of filtrate that's making it through the distal convoluted tubule. And the more filtrate that passes through here, the more of substances like electrolytes are also going to pass through there. And the more electrolytes there are, the more that are going to be pumped. The juxtaglomerular apparatus can actually sense that. So what these macula densa cells will do in response to an increase in those ions that are flowing through the distal tubule is they're going to release chemicals into the interstitial fluid surrounding these cells. So they're going to send a chemical signal to the glomerular apparatus. Actually, that signal is going to travel to the afferent arteriole. So they're going to signal through actually the release of ATP into this interstitial fluid. What they're going to do is signal the afferent arteriole to constrict. So high blood pressure, more filtrate flowing through the distal tubule, more electrolytes. The macula densa recognizes this and signals the afferent arteriole to constrict and to lower that hydrostatic pressure. So that's how you get a compensation in increased blood pressure. And if this persists for a long time, kind of jumping ahead a little bit to one of the hormones that regulates glomerular filtration rate is renin. So we talked about the RAAS, the renin-angiotensin-aldosterone system, for controlling blood pressure. And the kidneys, of course, produce renin. It's called renin because it comes from the renal system, the kidneys. So renin is going to increase blood pressure. And one way to decrease glomerular filtration rate is to decrease blood pressure. So the cells of the afferent arteriole create renin under normal circumstances that increases blood pressure. And aside from signaling to constrict or to contract the smooth muscle and turn down the faucet, they'll also decrease the production of renin, which would normally raise the blood pressure. But so if you have less renin, that's going to lower blood pressure and lower the glomerular filtration rate. So this juxtaglomerular apparatus basically serves as, on top of the myogenic mechanism, it's another sensor that allows the nephron to control its own rate of filtration. So there are two primary hormones that are going to affect the glomerular filtration rate. And one we've already mentioned is the RAAS, the renin-angiotensin-aldosterone system. And this will allow maintenance of a constant glomerular filtration rate when there's a dip in blood pressure, for example. So what renin-angiotensin-aldosterone system is going to do to increase the GFR in that case is going to be to constrict the systemic blood vessels, which will increase blood pressure, and also constrict the efferent arteriole. So what that's going to do in constricting the efferent arteriole is going to be clogging the drain, increasing glomerular filtration rate. It also is going to cause an increase in blood pressure by causing more sodium to be reabsorbed. And remember, water follows sodium, so the more sodium we're reabsorbing, the more water we're reabsorbing. And that will cause increases in blood pressure. That will again increase the glomerular filtration rate. Now, atrial natriuretic peptide is another hormone that's going to increase the glomerular filtration rate. And the way that it does this, ANP is of course made by the heart, but it's going to cause dilation of the afferent arteriole, so turning up the faucet, while at the same time clogging the drain, constricting that efferent arteriole. And what this is going to do is increase the GFR greatly. And with increased GFR, what that's going to do is going to speed up the rate at which filtrate moves through the nephron. The faster that filtrate that's going to become urine moves through the nephron, the less chance there is to reabsorb sodium. So the faster, the higher the glomerular filtration rate, the faster filtrate moves through the nephron, and the less time there is to reabsorb sodium, and the less time there is to reabsorb water. So that's increase in GFR is going to lower sodium and water retention. It's going to dilute the urine, and it's actually the heart's mechanism for decreasing systemic blood pressure. But it happens to do that through increasing glomerular filtration rate. So finally, in addition to hormones, the nervous system plays an important role in controlling GFR. And the way that it's going to do this is, one, directly causing vasoconstriction of systemic blood vessels. So constricting systemic blood vessels is going to increase blood pressure, increasing GFR. And the other way that it goes about doing this is that the sympathetic nervous system can also stimulate production of RAAS hormones, which will also lead to these effects. So directly causing constriction of systemic blood vessels and activating the RAAS are the neural pathways for regulating glomerular filtration rate. So that does it for our Week 2 YouTube lecture series, and be on the lookout for any correspondence in the near future.